Good morning, everyone, and welcome to Engineers House and Bristol. I'm Patrick Holden, Chief Executive of the Sustainable Food Trust, and Richard Young, who really is responsible for pulling this meeting together, has asked me to just make a few opening remarks. Of course, the theme is what role for grazing livestock in a world of climate change and diet-related disease.
That's the pointer. That's the advance. Okay, so the provocative title is Reducing Global Meat Consumption Would Improve Climate, Food Security and Human Health. So why is it not a no-brainer? So quickly over the next 20 minutes, Keith, if you can give me a 5-minute warning.
Yeah, okay.
I'm going to go over the evidence for some of this and then come back to the question about why is it not a no-brainer. Okay, that's not working, never mind, we'll do it like this. Okay, so delivering food security in 2050 will be extremely challenging. So this is looking at population projections going back to 1750. What you can see here is the population going it's about 3 billion in 1960. That had doubled by 1997. In October 2011, it passed the 7 billion mark. And when I looked it up last week, when I submitted the talk, it was 7.2 billion people. And you can see that it's not just a change in the total population, but also a change in where those people are living. So the vast increases in the developing countries. So that's the area globally which has the big significant impact on food security. And it leads us to an increase in demand. This is a slide that I found which shows the increase in demand for food over the next 40 or so years. It still knocks me off my seat to look at it, because the challenge over the next 40 or 50 years is to produce more food than we've produced so far in human history. That's absolutely staggering, isn't it? So we need to produce more food in the next 50 years than we've produced on this planet to date. That is the size of the challenge. Okay. So reducing demand for livestock products is essential. So this is my starting point. If we look at from a climate perspective, if we look at the greenhouse gas mitigation potential, this is not one that you're meant to read. It's just meant to show you some examples. This is collating together all estimates of greenhouse gas mitigation potential post 2007. So in 2007, the IPCC produced its 4th assessment report. Since then, we've had a few studies which have looked at mitigation potential. Agriculture is shown in yellow, forestry in blue. Forget the forestry for now, because we're talking about agriculture today. So at low carbon prices, we have this mitigation potential. Medium carbon prices, this mitigation potential. And at high carbon prices, this mitigation potential shown in this bar. That died as well. Never mind. I'll come and point at it. Shown in these bars here. This is just looking at the technical mitigation potential. What's shown over on the right-hand side is the mitigation potential that's available through demand-side changes. So that's through waste reduction. At the moment, globally, we waste about just over 30% of all the food we produce. And obviously, some of that is irreducible waste, but some of that is waste that we could reduce either pre- or post-consumer. And there are also changes in diet. So these demand-side options here, you can see, have a mitigation potential. These are technical mitigation potentials that are equivalent or perhaps even higher than the technical mitigation potential when you add up everything together, all the livestock feeding, improved management. So these demand-side options are under-researched and require more people to look at them in more detail. The reason diet is important is because there's a big difference in greenhouse gas emissions intensity between different products. So this is the greenhouse gas emissions in kilograms of CO2 equivalents per kilogram of products. And what you can see here is that extensive beef has a greenhouse gas footprint of around about close to 60 kilograms of CO2 per kilogram of product, compared to pulses and beans down here with a tiny emission per unit of product. So these are the basis of different life cycle analyses. I don't think many people would disagree that there are big differences between livestock products are non-livestock products, and within those the ruminants, because of the methane emissions from the enteric fermentation of digestion, have a large carbon footprint. So shifting a population away from these products to these products, if that were possible, you would reduce the greenhouse gas footprint of our diets. So just looking at the evidence for this in a little more detail, this was some lovely work that was done by Elke Steffest and colleagues back in 2009, and they had 4 different scenarios. So they had the reference scenario, which is business as usual. They had a no ruminant meat scenario, where all of the ruminant meat was removed from the diet and replaced with plant proteins. No meat at all scenario. A hardcore vegan one, with no animal products whatsoever, and a healthy diet scenario. So the reason to do this is not to say that these are possible futures, that nobody's suggesting that they are. It's just to look at the relative contribution of these issues. And what you can see is that the reference scenario has emissions of around about 3.3 gigatons of C carbon equivalents. The no animal products one is a third of that, 1.1 gigatons of CO2 equivalents. But we're not going to have no animal products, obviously. We're not going to have a totally vegan world. So there are intermediates in this. And if we just shifted people to a healthy diet, so we reduced overconsumption of livestock products, particularly in developed countries, but also those going through a nutrition transition into more meat products, we could reduce greenhouse gas emissions by about a third just by moving to healthy diets without asking anybody to give up meat or to give up their livestock and dairy products. Other studies have followed this. So this is Alex Popp and colleagues at the Potsdam Institute for Climate Impact Research. They looked at technical mitigation versus not technical mitigation, and then they looked at increased meat production and decreased meat production. So what you can see here is that if you've got no technical mitigation and you've got an increase in meat production, this is business as usual. This is what happens to the greenhouse gas emissions from global agriculture. If you use technical mitigation measures, you can reduce this a bit, so you can cut those emissions. But by far the biggest impact is from moving to decreased meat consumption. So by decreasing meat consumption, even without mitigation, you can have a vast influence on greenhouse gas emissions, and also show a decreasing trajectory. So all of the evidence is pointing in the same direction, that a change from by decreasing meat consumption, we can have a significant influence on the climate. And the last study, the last little bit of evidence, was a paper published by Bojana Bejel and colleagues looking at what would happen to each of these different categories. So a whole bunch of ecosystem services: cropland area, pasture, net forest cover, greenhouse gas emissions, carbon sink, fertiliser use and water use. This is what happens under the current yield trend. And you can see that all of these things are increasing. So the forest and grassland area are increasing, forest area is decreasing, greenhouse gas emissions up by 60%, carbon sink down, fertiliser use up by 60%, water use up by 125%. So we're currently in a situation where the agricultural food production system of the globe is creaking. That is going to creak more and more and more if we just follow the current yield trend. So what about sustainable intensification? What would happen if we can close the yield gap? If we can close that yield gap and make agriculture more efficient, we can reduce this relative to the current yield trend. So we can reduce the amount of, we can reduce the increase in cropland and grassland area, we can reduce the decrease in forest area, but they're still increasing. So it's better than the no yield one, but sustainable intensification gets us part of the way, but not all of the way. The last scenario is adding on to that yield gap closure plus demand options. So this is just moving people to a healthy diet and also reducing waste by 50%. So we're imagining that we could make those sorts of inroads. And what you see here is a decrease in the grassland area and the cropland area, an increase in the forest area, an overall decrease in greenhouse gas emissions, still an increase in water use and fertiliser use, but way less than in the other case. So this is another strand of evidence which suggests that on the basis of a whole bunch of ecosystem services, the demand side options, the waste reduction and the dietary change are absolutely critical for moving us forward to a sustainable future, and that sustainable intensification on its own will not get us where we need to be. Okay, so how will demand be met in the future? In the past, we've probably met this by expanding the agricultural area. Until we got nitrogen fertilisers, and our Rothamsted colleagues are here today, that all started off at Rothamsted. With industrial fertilisers, we were able to increase the per unit yields. But the suggestion is, and this is just qualitative rather than quantitative, that's not going to get us all the way. And here, if we're going to meet these challenges in the future, we're going to need some sort of demand side change. We're going to need to manage demand. The analogy that I would give is if you've got a screaming toddler in the supermarket saying, I want this, I want that, I want this, I want that, I want it, do you always give them what they want? And I would say, no, you don't always give them what they want. What you have to do is you have to manage that demand. And I think as a species on a planet with finite resources, we need to manage our demand for the products that are most environmentally damaging. Okay, there's another thing that I was asked to cover, and that's this idea that having livestock is great for the grasslands because they sequester carbon. Okay, and I think this will come up again. I think that's a myth. If you look at grasslands, this is a resampling study by Pat Bellamy and colleagues at Cranfield, where they looked at changes in soil carbon over the space of around about 25 years. And if you pick out the grasslands from that graph, you can see that grasslands in this country are actually losing carbon. So simply having a grassland, despite the fact that you can show from eddy covariance measures that they might be increasing carbon, when you go and look in the soil to see if they are sequestering carbon, in this study in the UK they don't appear to be. If you look in the long-term experiments like the Palace Leas in Northumberland and Park Grass at Rothamsted, you can see that soil carbon hasn't changed over many years. Park Grass has been running 160 odd years and there's been no change in soil carbon over that period. Other resampling around the world, this is going to the other side of the world in pasture systems there. Flat dairy pastures have actually lost a bit of carbon. There's been no significant change in grazed pastures for dry stock over those periods. So wherever we go and we look for the evidence of soil carbon increase, we don't find it. And we know theoretically that when you change out of for example, this is a cropland moving into a grassland, you will be in a phase where you will gain carbon. But as time goes on, this will steadily approach an equilibrium. This is data from Rothamsted. And as you approach that equilibrium, if you measure a change here, whilst you've changed the system, you may get an increase in soil carbon. But when you're on this phase, you've got a grassland that's remained a grassland, it's still a grassland, it's a flat line. You're not getting any increase in soil carbon over time. The other thing is if you just do a mass balance equation. So the suggestion from the eddy covariance measures is that grasslands are continually gaining about a tonne of carbon per hectare per year. If you just took a zero baseline in Roman times in this country, you would get a gain of around about 2,000 tonnes. So a tonne a year over 2,000 years is around about 2,000 tonnes of carbon over that 2,000 years. So many European grasslands would have stocks way, way higher than peatlands. Peatland stocks are around about 160 tonnes, so we'd be swimming in the stuff. There's just absolutely no evidence just from a simple mass balance equations that this is happening. It can't be happening. Having said that, if you improve the management on grassland, there is potential to increase it. So if you've got a poorly managed grassland, an overstocked grassland, an understocked grassland, changing the grazing land management can have a significant impact. In fact, it's one of because the grassland areas are so vast, it's one of the areas that has really significant climate mitigation potential. Thanks, Keith. So changing the grassland management is something that can be done. So if you've got a badly managed grassland and you move it to a better managed grassland, you can increase the soil carbon stocks. But simply having a grassland and saying, I've got one out the back, please give me carbon credits for it, is a non-starter. The reason I think this has happened is there's a confusion over carbon stocks and carbon fluxes. So carbon stocks is the amount of carbon that a grassland contains. They contain large amounts of carbon. So people say because they contain large stocks of carbon, therefore we have carbon sequestration. It's not the same thing. Carbon sequestration is the removal of carbon from the atmosphere. So if you've got a management change, over this time period, we get a big increase in soil carbon. The next time period, same time period, a smaller increase, same time period, a smaller increase, until you get to zero. So this is carbon sequestration. Carbon sequestration is large here. Carbon sequestration, even though we've got a high carbon stock, is zero here. The rate of change is zero. So a big carbon stock is not the same as a high rate of carbon sequestration. Okay, so grasslands under constant management do not increase in carbon. It could result from legacy effects. I won't go into that. So it could be the way that things have been managed in the past that could mean that we measure an apparent carbon sequestration. And we've got high carbon stocks, but that's not the same thing. But the policy implications are, and this is good for the livestock industry, if we can protect those high carbon stocks in grasslands, we're providing an ecosystem service in a way. We're protecting those carbon stocks. And that might be the way to go rather than claiming carbon credits for carbon sequestration, which I don't believe in. And if the management is suboptimal, it can be improved. So I'm not going to go into the human health angle, but if you're interested, Dave Tillman and Michael Clark, just before Christmas, published this paper in Nature, which is looking at the links between environmental sustainability and human health, and where they've looked at alternative diets, including vegetarian diets and Mediterranean diets. Dietary choices of individuals are influenced by culture, nutritional knowledge, price, availability, taste and convenience. So nobody's saying it's easy. But this diet health, environment health trilemma is a global challenge, and they're a great environmental and public health, of great environmental and public health importance. And showing similarly to the stuff that I've shown earlier, the lowest environmental impact are the meat-free diets, with Mediterranean diets somewhere between those and current diets. Okay, we might do this. I'm not going to dwell on this. We may do this by some mechanism of changing the tax regime. This was just Vesenius and colleagues in Sweden looked at if you internalise the environmental damage caused by different products, what would the tax be? And you can see that ruminant meat is way up there, so you'd have a large tax. If you were to do this, I think it's a very blunt instrument to try and influence behaviour like this. Matt's going to talk about behaviour change later, but it's a blunt instrument, and if you did do it, you'd have all sorts of social justice and equity issues associated with that. The poor in this country currently eat the worst, and this wouldn't help the situation. There's a whole bunch of other things to consider. So I'll just finish off on why is this not then a no-brainer? All the evidence is suggesting that dietary change is necessary for health, for the environment, so why aren't we doing it? The first one is that not all grassland is suitable for conversion to cropland. It's either too wet or too dry, and the best way to get human edible food from that land is via ruminants. But the problem is the concentrate feed, so you've got a big land footprint when you're feeding those concentrates. So that argues towards, even though grass-fed beef has higher emissions, in terms of per animal or per unit livestock product, because of this, then maybe it's still better than using concentrate feed to avoid that land footprint elsewhere. Food is an immensely social and culturally important and deeply embedded in all cultures and in our self-identities. People feel differently if they eat meat. People even feel in some cultures it affects, for men, if men don't eat meat, that affects how people perceive them as men. So, there are all sorts of really important issues, cultural and social, to do with social identities, that mean that meat is really, really difficult. There's resistance to interference in personal choice, and I think anybody saying we're going to put taxes on meat, that would be absolute political suicide. I can't see any mainstream politicians trying to interfere in personal choice to that degree. I think there's also resistance from the meat, livestock and dairy industries for obvious reasons. There are vested interests there, and possibly also in movements that rely on having a grass phase, so maybe some of the organic movements and maybe even some of the people in this room, there's resistance to it because it's part of what we do, and it's part of what we think is good and wholesome and healthy. So there's going to be some resistance there. Food taxes are a blunt instrument, and I think there's the opportunity for high quality grass-fed beef to fill a niche on an occasional luxury product is something that we should seize. So we could actually have a high premium on grass-fed beef in this country if we sell it as a luxury product that is occasionally, you know, we could get a high markup on that, and I think that's good for the industry. We do this really well in this country, and we've got great bioclimatic we've got great bioclimatic conditions in which to raise ruminants. So if we do it better than everyone else, we have a potential competitive advantage. So I don't think it's all doom and gloom for the livestock industry in this country. I'll leave the conclusions, I'll leave them up on there.
Thank you, Pete. Great start.
So I'm asking the question about ammonia and greenhouse gas emissions and who emits more. And you shouldn't read anything into the fact that my name is straight after that question. So to give an overview of the talk, I'll quickly go through the sources of those emissions, and then have a quick look at the current emission estimates that we've come up with. Look at the influence of management practices. I think that's really what we're about today, and some potential mitigations. So to begin with ammonia, which is an issue that probably doesn't get as much sort of airtime or publicity as greenhouse gases, but in my view is as important an issue. Ammonia is an important air quality pollutant involved in particulate formation, which has human health implications, but more commonly associated with issues such as eutrophication and soil and water acidification. And agriculture is the major source of ammonia emissions, and in the UK accounts for about 90% of total emissions. So here's a sort of pie chart of where those emissions come from, and you can see here that dairy and beef cattle, so the cattle sector, account for over 50% of total ammonia emissions from UK agriculture, followed by the other sort of livestock sectors, and then a fair amount from fertiliser use. So, you know, if we want to reduce emissions, it's the cattle sector in particular that we need to be looking at. And if we look at the cattle sector in a bit more detail and look at the sort of discrete sources of ammonia, we can see that livestock housing and manure spreading are the key or the major emission sources. So managed manure is the major emission source, and primarily ammonia is coming from the urine of those urine that's excreted by the animals. So while the animals are housed and we're collecting manure from those animals, the emission potential is in fact much larger than when the animals are grazing, when the urine rapidly infiltrates into the soil and is protected to a large extent from ammonia emissions. So to look at the influence of management on ammonia emissions, so here I've got some figures looking at the annual emission per dairy cow. And the UK average dairy cow emits something just over 30 kilograms of ammonia per year. So I just ran some scenarios through our model that we use to compile these emissions. So if we assume that all dairy cows in the UK were grazing all year round, I know it's a ridiculous assumption to make, but the emission would be far, far less, somewhere around 5 kilograms of ammonia per cow per year. On the other hand, if we assume that all those animals are all housed throughout the entire year, now there's various different kinds of housing. So initially, if we say they're housed on a slurry-based system, liquid manure system, then you see the emissions much higher. And if we assume they're housed on a farmyard manure or straw-bedded system, then slightly lower emissions than the slurry-based system, but both much, much greater than the grazing situation. So extended grazing systems will have lower ammonia emissions, there's no doubt about that. But, of course, there's potential there for increasing the other nitrogen losses associated with urine deposits in particular, going to grazed pastures. And there's a question of how efficiently are the manures used, or the excreta is used. If you collect it as a manure, you can decide when and how much to put onto the land and try to match nutrient availability with demand. When the animals are grazing, they're putting extremely high loadings of nitrogen onto the pasture, which is much more difficult to manage in terms of nutrient utilisation. So just some questions to address there. Moving then to greenhouse gases from agriculture, and we've heard a bit about that. So the main ones we're thinking of are nitrous oxide, which Ute is in fact going to talk a bit more about shortly, methane, both very potent greenhouse gases, with global warming potentials of sort of 325 times that of carbon dioxide. And again, agriculture is the major source of those emissions in the UK, 84% for nitrous oxide and just over 40% for methane. So if we have a look at where those emissions are coming from, and this is expressed as CO2 equivalents, almost half the emission from enteric fermentation by ruminants. 40% from emission of nitrous oxide from soils, so that's from nitrogen amendments to soils, and then the remaining sort of 11% there coming from manure management. So enteric methane emissions, obviously a major source there that we need to have a look at, and they are influenced by the production system. So again, this is looking at dairy cows. And if we have a look at large, high-yielding dairy cows versus small, lower-yielding dairy cows, so we might imagine these are your big Holsteins, these are your Channel Island breeds, and this is everything in between, so Ayrshires and so on. And the emission factor, the enteric methane emission factor, so kilograms of methane per head per year, is, of course, much larger for the big, higher-yielding animals. This is based using a model that we're developing, improving the inventory at the moment, the emission inventory methodology. But if we express that per litre of milk produced, it's in fact the reverse. So the big sort of, you might say, highly pushed animals, in fact, have a lower emission intensity per litre of milk. Now, I must stress, this isn't life cycle analysis, this is just the emission, the enteric fermentation emission within the farm gate, if you like. So we're not talking about where the food's coming from and all the other sort of energy issues associated with that, but purely looking at the emission from the cow itself. The emission intensity is lower for the bigger, higher yielding animals. So what else influences the enteric methane fermentation? If we try to pull that apart a little bit more between the milk production and the live weight of the animals. So live weight itself, it doesn't have a huge influence. So we've got the small animals here coming out about 115 kilograms per head per year, and the much bigger animals just over 120. If we look at the feed those animals are getting, it's quite often said that we should be thinking about what we're feeding animals and how much that influences the methane emissions. Now here I've just looked at changing within typical standard feeds. This isn't looking at alternative feeds that may lead to emission reductions, but if we change from predominantly concentrates to predominantly grassland, so predominantly concentrates here, predominantly grazed grass here, in fact it makes very little difference to the per head methane emission. Whereas milk yield, if we look at an animal that's producing about 5,000 litres a year to an animal that's producing 10,000 litres a year, that does have a very big multiplier. But again, as I said before, per litre of milk produced, in fact the slope would be the other way. So depending on the production system, we can influence to a certain extent the enteric methane fermentation emissions from the animal. So diet and body weight seem to have a relatively small effect though, and production level has a much greater effect. Of course, there's a whole range of other considerations that we need to take into account, and things such as the longevity of the animal, the fertility, so how many replacements you need per milking dairy cow, the overall health of the animal. All of these will also have an influence, and I think some of those issues will be discussed later today. So looking at some potential mitigations, first to look at the ammonia losses and nitrous oxide losses. So one way of reducing those emissions is to try to reduce the nitrogen excretion by the animal. And we can do that by lowering the nitrogen intake, so modifying the diet of the animal. So if we've got less nitrogen going into the animal, less nitrogen in the excretion, and then as that manure is managed, either directly going to the pasture or going through the house and the manure storage and to the soil, so the emissions of ammonia and nitrous oxide will be lower from each of those stages. So it's a very kind of efficient way of reducing emissions throughout the whole system. And many people have shown this, and this is a study we did, laboratory study we did some time ago, looking at the difference between a low crude protein diet and a high crude protein diet for dairy cattle, or a diet with increase in amounts of condensed tannins, which to some extent protects the nitrogen from the rumen, and it's better utilised in the hindgut. And very clear differences. So much lower ammonia emissions from the subsequent manure for the low crude protein diet, and also lower ammonia emissions as we increase the condensed tannin content of the diet. But there's questions about, well, how low can we go with protein content before we start to influence the animal production, the fertility, and so on. So in fact there's a DEFRA project led by Reading University that's looking at that just now, looking at a group of animals over a long term, a number of years, to really see how far we can reduce that crude protein level in the diet. And also questions about how can we achieve this in a largely forage-based system. It seems to me it's much easier to sort of tune diets in a house situation for cattle, and less easy under a forage grazing or forage-based system. But again, I think those are questions that may be addressed later on. In terms of emissions from manure management, in particular ammonia emissions, and one of the easiest sort of mitigation techniques that's been out there for some time now is to use low emission spreading technology. And there's a number of different ways. So band spreading, putting the slurry into narrow bands on the pasture, injecting the slurry into the pasture, or with slurries or manures, rapidly incorporating those into the soil where there's an opportunity to do that. And these will give very large reductions in ammonia emission. There is a potential to increase nitrous oxide, but this isn't seen consistently. And I think if the manures are applied at the right time and in the right amounts, then the risk of increasing nitrous oxide is low. And finally, another example of mitigation, the use of nitrification inhibitors. And these are very effective at reducing nitrous oxide emissions specifically. So this is, it's difficult to read that from the back, but this is nitrous oxide emissions from urine applied to soil, so from a urine patch, for example. And you see after the urine goes to the soil, a very large spike in emissions. The green line is using a nitrification inhibitor, DCD, and you can see there's virtually no emission there at all. And we've done a number of measurements of this and shown that by and large the DCD is very effective at reducing emissions from urine applied to pastures. There's some influence of particularly of the soil temperature, and the efficiency is lower at a higher soil temperature. So very effective, but there's a cost associated with that. There's limited evidence of any yield benefit, so why would a farmer use nitrification inhibitors? And there's also a question about how you're going to use that in a grazing situation. If you have to spray the DCD onto the pastures every so many weeks, again, that's a cost to the farmer, an extra operation. So more research needed around some of those issues. So finally, to come to the summary and conclusions, cattle farming, no doubt about it, is a significant source of ammonia, nitrous oxide and methane. It's inevitable. There's going to be some emissions. So the question is, how big do those emissions have to be? Ammonia emissions primarily from the managed manure, methane mostly from the enteric fermentation, and nitrous oxide mostly from the soils. So three different areas to look at there. Management will influence those emissions, but there are potential trade-offs. If we change the management, some of those emissions may increase, some may decrease. There are some good mitigation methods out there, particularly for ammonia. I think we're still looking for some real good ones for greenhouse gas emissions. And I think I mean, we have to accept that there's always going to be a variety of different production systems. As Pete said, not everybody in the world is going to be a vegan. Not every farmer will manage their cattle in the same way, and there's good reasons that will happen. But we need research to improve the nutrient use efficiency and to give us mitigations within all of those systems. And I think, speaking from a Northwick perspective, particularly, I think there's more scope for precision in grazing and grassland-based ruminant systems. So I'll finish there.
Thank you, Tom. Our next speaker is Dr. Ute Skiba. Ute I've also known for some time because we've worked on similar topics. Ute is a biogeochemist at the Centre for Ecology and Hydrology, and she leads the group there on land-atmosphere exchange of greenhouse gases. Ute, thank you.
Okay, right. So I'm going to talk about nitrous oxide, specifically from grasslands. And just some facts on nitrous oxide to remind you. It's the long-lived greenhouse gases, over 100 years in the atmosphere. It's responsible for 10% of the total global anthropogenic radiative forcing. And now, since we don't use the CFCs much anymore, it's the largest source of stratospheric NOx, so responsible for the ozone hole. And 77% of nitrous oxide is produced by microorganisms in the nitrification and denitrification processes, which occur everywhere in the world, tropical ecosystems, Arctic ecosystems included, and in waters, oceans, rivers, soils. Okay, these are the global sources of N2O. They're mainly, so the largest sources are the oceans, natural soils, and agriculture is responsible for about 16% of total global N2O emissions. However, the atmospheric increase in N2O since the Industrial Revolution is responsible due to agricultural activity. And the same is true for methane. The atmospheric increase in methane is responsible to increased fossil fuel use and animal rearing. Now, in the UK, agriculture is responsible for 10% of the UK greenhouse gas emissions. And that's the 10% of the UK greenhouse gas emissions. All the sources are over there. Over 80% of the UK N2O emission is from agriculture, mainly from soils. 44%, I think Tom mentioned that already, 44% of the methane, of the UK methane emissions, which is basically livestock and then waste, landfill sites, and only sort of less than 1% due to sort of combustion, tractor use, which is only 0.9% of the, well, less than 1% of the UK total sources. So, what are the agricultural sources of N2O? So these are the categories. So it's mainly if you put fertilizer on your soils, manure your soils, use legumes, nitrogen in residue, leaching is a big one. And so there are many different categories which give rise of direct or indirect N2O emissions. Now, grasslands are in particularly good sources of N2O because N2O is basically the processes, the microbial processes, nitrification, denitrification. They don't occur all the time. They need nitrogen as a source, a food source for the microbes, but they also need the right conditions. There needs to be just the right amount of oxygen or lack of oxygen inside the soil. So you can sort of assume these processes just occur, they're just opportunistic, they just occur when the conditions are right. And it's just like a sort of lots of flashing lights, which is sort of activities here, next minute activities there. So spatially and temporally, very, very heterogeneous. Because of this demand of low oxygen, it's the high N2O emissions tend to be dominated in the high rainfall areas, especially the grasslands in the western part of the UK. And those ecosystems that have large and frequent, they also have high inputs of nitrogen, and there you would have extra compaction and then the urine and dung patches from the livestock. And now I will give you an example of our own work where we have studied for the last, well, since 2002, whoops, what's that? We studied the greenhouse gas budgets at an intensively grazed grassland just outside our institute in Midlothian, south of Edinburgh, where we've measured the carbon dioxide, the NEE, methane, nitrous oxide by various methods, the traditional chamber methods for the N2O and methane, but also with eddy-covariance methods. So our work there, our very intensive measurements through various research programs by EU, DEFRA and NUC funding has provided us enough data to provide very detailed carbon and nitrogen and greenhouse gas budgets for this particular site. Okay, I'm just sort of showing you some of the measurements just to give you a feel for it. These are sort of chamber measurements of nitrous oxide. That's for 4 years, and that's just an average of 4 chambers. I mean, the chambers measurements are very spatially variable. But all these arrows are when we fertilize the field. Sometimes you get an immediate fertilizer response. Sometimes you don't get any. And that's very, very typical because you have to have the right rainfall at the same time in relation, the right moisture content and the nitrogen together to produce your N2O. Which means the emission factor, the percentage of nitrous oxide lost as, the percentage of nitrogen lost as N2O varies very much between the years. These are our net ecosystem exchange measurements of CO2 for CO2, and this is a comparison of, again, this is just for 4 years. There's sort of half-hour measurements. You get uptake in summer periods. The blue line is the intensively grazed grassland Easter bush. Ochenkorth moss is the drained peatland. And for the intensively grazed grassland, you have larger net ecosystem change of CO2. The interannual variation depends really very much on the length of the growing seasons. Methane fluxes, we did actually use eddy covariance to measure methane emissions from our livestock on this field. And that's just sort of to show it basically depends on how many animals are within the fetch of the analyzer. And our measurements sort of calculating up to the total was very much in agreement of the IPC published emission factors for sheep, which was good. So we put all this stuff together and produced a greenhouse gas budget for Easter bush from 2002 to 2010. This paper should be published very, very shortly, I hope, now. So basically, you've got your net ecosystem exchange of CO2, sort of apparent carbon sequestration, which is offset by your N2O emissions, which vary depending on the rainfall, when you add the fertilizer, and your methane emissions, which are influenced by the number of animals you have. So on average, about 60% of the NEE sequestration is counteracted by N2O and methane. NEE isn't the carbon budget. You have to include also everything you take off. You have to take the methane emissions from enteric fermentation, excretion, soil, from the organic fertilizer you apply, the leaching losses, which we usually don't measure, we often don't measure, the dissolved organic carbon we measure. So that's, and there's sort of modeling data. You need to know how much carbon is in the wool meat harvest, lots and lots of assumptions. It's very difficult to get it right. Apparently, this still suggests that there is a net carbon sequestration of minus 180. However, there are two ways to study carbon cycling. The other way is to actually look at the carbon stocks in the soil. And in 2004-2011, for this 5-hectare field, we took 100 cores 60 centimeter down and measured the carbon stocks in them. A huge amount of very hard physical work and quite expensive. But when we've done that, we found that the carbon stock change was about 9 grams of carbon per meter square per year, and not this 180 which we sort of measured with the net sea sequestration. As you can see, the error bars are massive, and we've come to the conclusion that basically it's zero. This is not because of the large differences, uncertainty. So basically intensively managed grazed grasslands in southeast Scotland on this clay loam soil is definitely not a carbon sink. And I know other people are probably sitting on similar data. I think we need to sort of do some more work there. So because of this discrepancy, we didn't know which method is right. Our paper has been sitting there for the last 2 years and hasn't been published yet, because we wanted to make absolutely sure that our calculations are correct. We feel they are now, and this is our conclusion. So, I've said that. So basically, it is very unlikely that the NO and methane emissions from intensively grazed grasslands can be sort of sequestered by CO2. And occasionally, you do have to plough this field, and then you will lose some more of the carbon. So we need more data, more work. I don't think I need to go really into this, but have I got 1 minute? Yeah. So can grasslands actually be carbon neutral? There's this paper by Jean-François Susanna and Gilles Marek or whatever. And they basically say, sort of, this is the modeling exercise mainly based on some NEE data. So extensive grazing can increase soil. So basically, very extensive grazing would actually stimulate the soil nitrogen cycle and carbon sequestration. So they actually wanted to sort of find out, is there a tipping point to, what is the tipping point that where you have sort of sequestration without too much NO methane emission systems? And and they want to sort of also now ask this question, how can one use management to minimize the NO and methane emissions and increase sea sequestration? But they are not really suggesting that in an intensive situation you could ever sort of reduce these to carbon zero situation. Thank you.
Thank you.
I should draw attention to my co-conspirator here, Johnny Johnston at Rothamsted, as some of you'll know. If you believe it's possible, he's even older than me and knows much more than me. He's an enormous source of knowledge about data in our long-term experiments, so you'll see his hand here quite a bit. We were asked to talk about soil carbon under mixed cropping, which I'm interpreting as layerable systems. We couldn't stop ourselves just straying out of our remit a wee bit to get some general pros and cons for a mixed system with layerable compared to just arable farming. There's plenty of pros. You can read them quicker than I can read them out, so I won't bother. Things like recycling nutrients, so on. Lots of sensible things to having a more mixed system. The cons are really practical and economic issues, really. Johnny was very keen to point out the cost of fencing. If you've got animals, you need more and better fencing than if you don't. There's all sorts of things that work against it. Now, you know, the point was made that economic to some extent are a human construct, so that perhaps could be rigged differently. Anyway, Okay, we need to worry about the state of soil. I like the phrase Earth's living skin, and we're in International Year of Soils and so on. A big factor in affecting how soils are and their quality, if you like, what they do is the amount of and type, if you like, of organic matter, organic carbon in soil. And this really has two impacts. One is on the quality of the soil, its soil physical properties and how that affects the crops growing in it, and also, as Peter's commented, of course, in the sort of climate change area. Now, the amount of organic carbon in a soil, as has been touched on before, is a balance between inputs and outputs, and it tends to move to an equilibrium. So the inputs are mostly things derived from plants, whether it's roots or above ground inputs, plus any manures and the like that are added. The outputs are mostly decomposition of organic matter by soil microbes, usually, well, mostly to CO2, but to methane in anaerobic situations. But dissolved organic matter, as Ute mentioned, is also an output. And so can soil erosion be. How you interpret that in a climate change context is sort of interesting, and I'm not going to go into it, but we might want to discuss that. Let me give you, first of all, an example of these sort of changes in a sort of a fairly extreme situation. This is an experiment at Rothamsted where we started, and I didn't start, maybe Johnny did, in the 1940s with two sites. Whatever. One started as grassland, it had been grassland for a long time, one had been arable for a long time. The grassland one, this is the soil carbon in those sites over the next 60-something years. The grassland, the management of that grassland was a bit more intensive from the start of the experiment before, so its soil carbon went up a little bit, but is now topped off, if you like. The bit of it that was ploughed up and went into just arable farming, it's come down, and within about 40, 50 years it sort of bottomed out at a low level. The one that started arable was already low because it had been cleared from forest and whatever way back over there hundreds of years ago, if it stayed arable, it stayed bottomed out, didn't go down anymore. It had reached an equilibrium. It went into grass, it went up, and it's on its way to joining the very old grassland. But that is an extreme. Here's a slightly more subtle effect in what's called the woven layerable experiment, where there are combinations of grass and arable cropping. Slightly complicated. It's an experiment where there are 3 treatment cropping, years of treatment cropping, followed by 2 years of arable test crops, as they're called. The treatment cropping is either arable, so you therefore get a continuous arable at one extreme, or the test cropping is 3 years of a grass lay given nitrogen fertiliser, or 3 years of a grass clover lay, so the biological fixation going in. And yields have been measured on this and so on, but also soil carbon has been measured over this period, well, 60 years. It's a sandy soil. And if we compare the continuous arable here, soil carbon, with the organic carbon in the lays, and there are humps and bumps and some complications in the past, look at it about 2000, we can see that the organic carbon expressed as a percentage increased by about 0.2 something percent. But that difference was pretty much established within 30 years. So if I work that out in terms of the stock of carbon changing, it looks like the stock of carbon there under lay arable compared to continuous arable has increased by about 6.8 tonnes per hectare in 60 years. I think that's a generous reading off the graph, but let's be generous for a moment to make us happier. That difference was established in 30 years, divide the 6.8 by 30, and we get something like 0.2 tonnes of carbon per hectare per year, as if you like, as a carbon gain from having a lay arable system instead of just arable on that sandy soil. There's a similar experiment at Rothamsted. I haven't got a nice graph of it, but these are taken from a comprehensive review by Johnny and colleagues in Advancing Agronomy some years back. And here on this silty clay loam soil, data after 36 years, similar thing, a 3-year treatment cropping, continuous arable or lay phases, but then 3 years of arable test crops, and they worked out that the increase in soil carbon were 0.3 something and 0.2 something percent in that period. And if you work that out again to changing carbon stock, the rate of change, somewhat surprisingly, is about the same as on the sandy soil. But they're very much rounded off, and there's all sorts of, we're looking for small changes against big numbers really here. Now I then did a very crude sort of sums, which sort of thing that Pete and I did more than 20 years ago when we first worked together, multiplying small numbers by big areas of land, but I did it anyway. To hell with it. Yeah. I took the arable area in the UK, multiplied that by this 0.2 tonnes of carbon increase if we converted all arable land to mixed lay arable, and then I converted it to CO2 and compared that to UK greenhouse gas emissions, the basket of greenhouse gases, and if you like, the annual benefit from having, or the, if you like, the avoided emissions from the lay arable system works out at about 0.6% of total UK greenhouse gas emissions. Now I think that's a pretty generous number. I'm surprised it's as big as that actually, and I think there's all sorts of reasons why it might be a bit too big, but there you are. So there is a little benefit, I think. I don't mean little, I mean a little benefit in terms of sequestering carbon if you went from continuous arable to a lay arable system. However, maybe more importantly is what that little bit of extra organic carbon does to soil physical properties, and I want to demonstrate that that can be interesting and useful with some experiments on straw. I haven't got the data for lay arable, but we did a review a few years ago of experiments where straw, cereal straw, had either been returned or taken away. There were experiments going on between 6 and 56 years in various parts of the world. And there was, as you'd expect, a trend for an increase in soil organic carbon if you return straw rather than put it away. But the effect was incredibly small. And the increases were actually only significant in 6 out of the 25 experiments we looked at. The little trend was there, but not quite reaching statistical significance. However, in some cases, the authors had measured what we might call active fractions of organic matter, such as microbial biomass, and these, where they had measured them, had changed proportionally much more than total soil carbon. So I think, and these have got interests and importance of their own. In some cases, in these 20-something experiments, the authors had measured physical properties such as aggregate stability, penetrometer resistance or water infiltration rate. Where they had measured them, they were always greatly improved in the straw return treatment, even where you could not measure any change in total soil organic carbon. So there are subtle, the small changes have big effects, is what I'm saying, and there's some similar results I've been looking at recently in Africa and South Asia for things we do under what's called conservation agriculture. This, of course, has got implications for potential implications for crop growth. Time is short, so I won't show you the evidence, but I've got it here. On the Rothamsted long-term experiments, we have one on winter wheat, the Broadbalk experiment, and one on spring barley, the Hoosfield experiment. There is evidence from these that there is a positive impact of having a higher organic carbon content from manure applications if you've got the short duration crop, the spring barley. It's much more sensitive to soil physical conditions, it would appear from our data. Whereas the long duration crop, the winter wheat that's there nearly the whole year, seems to overcome poor soil conditions much better. Maybe on other sites it will be different. But there, in some cases, extra organic matter in soil is going to be a benefit that can't be totally reproduced by nutrients. If we have got improved soil structure from greater organic matter content, as we would have if we had more mixed lay arable type farming, this means that roots can explore the soil better and possibly absorb water better, but also more efficiently capture immobile nutrients such as phosphate. I won't dwell on this, but again, this is some data from Johnny. This is looking at this Hoosfield experiment again in the comparing high and low organic matter treatments. Spring barley has been grown, nitrogen rates have been applied. These show you the best yields they could get when they applied nitrogen, so that was not limiting. But where you'd got poor soil structure from the low organic matter, you needed a high concentration of crop available P. I missed out, this should be milligrams per kilogram here. You need a lot of P in the soil. Where you've got better soil structure, you could get the same or higher yield, but with a lower concentration of P. So the better structure gave you the potential for more efficient utilisation of other nutrients. When they destroyed the structure, did a pot experiment, it didn't make any difference. Okay, final comments. What happens if we go from arable to mixed arable grass cropping? Well, there are some clear benefits. There is a small increase in soil organic carbon compared to continuous arable, and that's got a small climate change benefit for some years, of course. If Pete explained exactly, it tops out, and after some decades you get no more. But let's ignore that bit for a short time anyway. I would worry that there is a risk, it seems to be, if we had more of this layerable sort of system, perhaps there's a risk that some current grassland soils would be ploughed out because we might end up short of arable, and that would counteract the benefits that we're going to get in the arable going into grassland sometime. So I would worry a bit about that. A real benefit will be that if we have these mixed arable systems, particularly if you've got clover in the pasture phase, then there's potential for using less nitrogen fertiliser when you grow the arable crops. So that's beneficial from many points of view. Improved soil structure if we've got more soil organic carbon with agronomic and environmental benefits, less erosion, etc. And of course more diverse landscapes and habitats. But there are some buts. If you have that sort of system, of course, in the years when you're growing your pasture phase, you're not growing arable crops. So your production over an area, a country or whatever, of the arable crops just has to be less. Now, that's where the demand things come in, but we do have to take it into account. Don't just look at the yields in the year straight after, in the 1 year straight after ploughing up a lay. You've then got to think, what do we do with that pasture? And in order to do it, of course, there need to be very large changes in the agricultural structure that we have. Now, that may be possible, but to make it economically feasible would be challenging, but it's certainly something we shouldn't exclude it. So thank you very much. And of course, as a good member of the British Society of Soil Science, I have to plug the fact that in, I don't know whether I'll be there then, but we host the World Congress of Soil Science in Glasgow in 2022. Thank you.
Thank you, David. Well, we're making excellent time. So our next speaker is Professor Richard Evershed. Richard, over to you. Richard is Professor of Biogeochemistry at the University of Bristol and Director of the Bristol Biogeochemistry Research Centre. And he's going to tell us a little bit about this methane sink in soil.
Okay, well this is a slight change of tack, but I think David started to sort of hint at the importance of getting into some of the mechanisms that are involved in some of these processes. I'm not I don't do inventories. There are other people in the room that do those much better than I do, and I don't do modelling. So what I'm going to show you is an example of how we start to probe some of these processes involving greenhouse gas cycling in a little bit more detail. And yeah, the grand title of my talk was Richard's invention, because I wasn't quick enough in getting back with an email. So this is the real title of my talk. It's about a technique called stable isotope probing and how we're using this to probe methanotrophic bacteria, so probing the soil methane sink. And oh, hello.
It's not working very well, Richard.
Okay. So where I'm coming from is that what I do as a chemist, because I'm a chemist, is I'm using organic molecules produced by the plants and the organisms that are associated with the soils, to try and get insights into global processes. So what I'm doing is going down through scales to the sort of microscopic level to look at processes that are essentially invisible to the naked eye, and using molecules as proxies for those processes. So that's what I do, and that's what we've been developing for about 20 years in Bristol. And we stumbled across about 15 years ago the idea of bringing together that idea of using of molecules to actually starting to address the question of the of studying the methane sink. And I don't need to go through all this. A lot of this is a repeat, but you know, the disturbing thing that sort of hit home to me at the time was that just how methane is the gas that's sort of increased in concentration so massively from pre-industrial times in terms of its the relative increase to pre-industrial times. The sources are being discussed extensively. The sinks become important, and these seem to be sort of little talked about. The main sink of methane is its reaction with the hydroxyl radical in the atmosphere, which is believed to account for about 90% of the loss of methane from the atmosphere. But soils play a role in looking after methane concentrations to a degree by the methane oxidizing bacteria that live in them. And The soil sink is estimated at about 5%, but I suspect that that is a rather poorly understood figure. So I'll talk a little bit more about the sinks in a moment. Why would we be interested in it? Well, that 5% is manageable. We can't really do anything about the atmospheric sink. We could manage it in that sink in soils by perhaps changing some of the ways we use our soils, fertiliser effects, tillage, etc. So there are two different types of methanotrophic bacteria. There are these low affinity bacteria. These are the high capacity methanotrophs. These are ones that live in wetlands. And these are extremely important organisms. Basically, in these wetland systems, you have methanogenic archaea that are producing large amounts of methane, and these methane oxidizing bacteria basically provide a protective shield where most of the methane is actually oxidized to carbon dioxide, with only a relatively minor proportion escaping to the atmosphere. These organisms have been cultured in the laboratory, so they've been extensively studied, and they're shown to mediate this process. And it's the process whereby methane is taken through methanol, formaldehyde, formic acid, and converted ultimately to carbon dioxide. And that's a green process because you're taking methane, which is a more powerful greenhouse gas molecule for molecule than carbon dioxide. But also what they do is they capture the carbon from the methane at that formaldehyde step, and they use it as a source of carbon to grow new methanotrophs. So we sort of know how the biochemistry of methane capture works from this. Interestingly, I think what's somewhat underestimated, someone else has done this calculation, this biological sink that's protecting us is probably about the same size as the atmospheric sink, if you think of it as a biological sink. And I think that is often an underplayed term. So this is why wetlands are so important in their capturing of methane. So, but the one I really want to talk about, which is relevant to agriculture, these are the high affinity methanotrophs. These are low capacity methanotrophs, and these live in well aerated soils, grasslands, forests, agricultural land, and these consume methane directly by drawdown from the atmosphere, and they're surviving on these trace atmospheric levels at this sort of 2 ppmv sort of level. But they are an important sink of methane. They're completely rather perplexing because they've never been cultured in a laboratory as far as I'm aware, but they're all the while converting methane to carbon dioxide and capturing methane for their own growth. So they can't be cultured in the lab. So how do we study them? Well, their activities are mostly studied by making flux measurements, and so that's an indirect way of telling whether you've got a methane oxidizing community. Another way in which people like myself and other soil scientists study unculturable organisms is to use culture-independent methodologies. And one method that's used is this phospholipid fatty acid analysis. And what this relies upon are the phospholipids, which make up all the cell membranes of microorganisms. They form the biolayer that then packages all the other genetic material and other biochemical apparatus. And the principle that comes into play here is that phospholipids, which make up these membranes, on cell death, these are extremely short-lived biochemicals and are quickly recycled back into the system. So if you do an extraction of phospholipids from the soil and measure the phospholipid fatty acids that make up the membranes, then that is a proxy for the living organisms that are in the soil at the point at which you did the extraction. Okay, so it's a very simple analysis to do. You just extract the phospholipids and measure the phospholipid fatty acid by gas chromatography, and you have a proxy for the living microbial community. It's a sort of crudish proxy, but it works quite nicely and allows you to assess the impacts of various management strategies on soils. So that's what we do. And this is the GC chromatogram of the phospholipid fatty acids from a soil. And what you get is a lot of different phospholipid fatty acids of different structures, which we get by doing mass spectrometry work. And you can crudely start to assign some of these structures to particular types of microorganism that are known in soils from reference collections, from reference databases. But unfortunately, none of those phospholipids is diagnostic enough that we can say it comes from a methanotrophic organisms. So this type of idea completely fails for studying this methane sink. So, what do? Well, I'm a fisherman. And what we have to do is get a bit cleverer. If you're going to catch big fish, you've got to get the bait right. And there's no need to lie when you get to this side. We need to get the bait right. So what we did was start to realise that we have a bait for methanotrophs. It's a very convenient one. And you can buy, off the shelf, you can buy 13C labelled methane. So this is isotopically labelled methane. It has just methane with one extra neutron. You can buy it. And it opens up a whole way of studying these otherwise unculturable communities at high specificity. So you have the idea that you have a range of microorganisms in the soil. There's a mixture of methanotrophs and lots of other types of microorganisms. You expose them to 13C labelled methane. Only the methanotrophs will consume the methane, and they will become 13C labelled. It's a very simple idea. So the only challenge then becomes of how do you detect those methanotrophs amongst this otherwise excess community of other organisms. Well, we do that by, well, this is how we do the incubations. We literally have synthetic air made up, and we have a chamber which we put our soils in, and we let the air flow through the chamber, and over a period of sometimes up to months, these actually are quite slow-growing organisms, they gradually accumulate the methane, and you can then extract the phospholipid fatty acids and do the analysis. We then use a technique called gas chromatography isotope ratio mass spectrometry. I shan't go into it, but it's a technique that we've been great promoters of in Bristol, and this is becoming quite widely used amongst people undertaking this type of process-based biogeochemical research. But what you see, these are basically what we call the carbon isotope value. This is the value, carbon isotope values of fatty acids where there is no methane uptake. And what you can see, these are fatty acids on that chromatogram that I showed earlier, which are showing higher levels of 13C incorporation. So these are fatty acids that have been made by those organisms living in that soil. What you can do is you can convert these numbers here to absolute concentrations, and you get histograms which you can start to match to the profiles that are seen in known cultures of the culturable organisms, and you can start to do some crude chemotaxonomy and saying what types of organisms these are. These happen to be a group called type 2 methanotrophs. But what you can also do, you can start to do some calculations that tell you just what the population of those organisms are in the soil. You can do a crude conversion to cell numbers, and they are present at vanishingly low concentrations, of 0.01% of the total microbial biomass are these methanotrophic organisms. So important methane sink, but very low concentrations. Okay, yep, fine, no problem. So this is a study that we've sort of piggybacked on, which I think will illustrate how you can sort of use this in an agricultural setting. And this was my postdoc, Pete Maxfield, who did a lot of this development work. And he stumbled upon this experiment from that was being, what was then the Institute of Grassland Environmental Research experiment that was being run at Bruneth Moor up in the Brecon Beacons. A very nice experiment where they're actually looking at the effect of, I think it was a sort of upland biodiversity on from various grazing treatments, and they divided the hillside up into various treatments so they could plots so they could do different fertilizer treatments. But we piggybacked on to see what was going on below ground. So just to show what happened, so we picked out 3 treatments. There was a no fertilizer treatment, a nitrogen, calcium, phosphorus and potassium treatment, and then the same treatment but without the nitrogen. So these are the total phospholipid fatty acids, high concentrations of bacteria, and these are the concentrations of the PLFAs then following the methane incubation. So these are the methanotroph PLFAs. Concentrations, as I say, very, very low abundances of cell numbers. But what you can see, the no fertilizer treatment, we get the highest abundance of methanotrophs. As soon as you start to add inorganic fertilizers, you're suppressing the methanotroph population by anything up to around about 80%. So this is an extraordinarily widely used treatment, which is obviously impacting on this methane sink probably everywhere. This phenomenon has also been seen at Rothamsted and I think this is something that sort of should be some cause for concern, but the paper we published has hardly ever been cited, interestingly. So just to finish, so I hope I've shown you how this stable isotope probing allows us to link the actual methane oxidation to the actual organisms involved in doing the oxidation. They're present at very slow numbers. The fertiliser additions definitely do suppress the oxidation, and work at Rothamsted has also shown that tillage is also disruptive to these organisms, and the cell numbers there where we've done measurements are down at 10 to the 5 cells per gram of soil, whereas 10 to the 6 in the grassland, so they're completely and utterly knackered there. I think we need more work to understand the potential capacity of the methane sink nationally and globally. If you think we've gone through several phases of agriculture, forest clearance would have caused probably one drop in this methane sink capacity, and then the development of modern agricultural methods has probably caused another impact on the sink, and I suspect we have no idea what the soil methane sink capacity really is. And we have some methods which we use N15 isotopes to look at nitrogen fertilisers, but that's for another day, and I thank the BBSLC and the NERC for funding this.
By the way, the knackered soils at Rothamsted have just produced the highest yields of wheat ever on that experiment, so they're not knackered. Exactly. Thank you, Richard. Richard Young wanted to make a few comments before we break for coffee.
Thank you. Well, as you know, I'm not a scientist, but I've given myself just a few minutes to put a few things on the table from our Sustainable Food Trust point of view, really building on the comments, first of all, that David Powlson made earlier on. One of our growing concerns is actually about the sustainability of soils globally. And Pete Smith made some very important points about the fact that there's a lot of controversy about whether whether grassland soils are even holding their soil carbon. I think we'd argue that probably that's a lot to do with the way they're being managed, but just leaving that to one side, I think what we could be clear about is that in soils which are just being used to grow crops, you will always see a significant fall in carbon. And not just carbon, soil organic matter is only 50% carbon. You're seeing a loss of nitrogen and other nutrients too, and particularly you're seeing a loss of soil structure. Globally, the United Nations has told us that 51% of all soils are either severely or moderately degraded now. And there are predictions that at the current way in which soils are being used, there's really only, in layman's terms, there's only enough soil left globally for another 60 harvests. So a huge, huge problem in the way that soils will be managed. And the problem, from our perspective, with a concept that we can just grow crops and not have animals and not use those animals and grass to help make the land management sustainable, is that actually this is how the vast majority of the carbohydrates that are now being produced worldwide in prairies of South America, the prairies of North America, the prairies of Russia and Australia, where we're seeing, even here, I think it's probably soya, so it's a crop which is fixing nitrogen, but all the studies show there's a massive continual loss of carbon and structure from these soils. And we have to ask, how long is that sustainable? And how can we make those soils sustainable if we don't actually start to use grass and livestock in those rotations? That really is building on these points. The question I'd leave to ask is there any way of having a continuous arable cropping system which is sustainable without using grass in the rotation? Now, the next point I want to put on the table is about nitrogen. I've had a couple of mentions of that from Richard about the impact on the methane sink, and Bob Rees is going to talk about legumes in a minute or two after the break. But we do have to remember that to produce food, and we all depend on food, we need an adequate supply of nitrogen to farming. There's only really two major sources of that. One is nitrogen fertiliser, the other are legumes. And we've now got the European Nitrogen Assessment and a number of other studies which have actually shown us that if you take the full range of negative impacts of nitrogen fertiliser on society, society itself is paying about 3 times as much as the commercial benefits that farmers are actually getting from using nitrogen fertiliser. We also have to ask, how sustainable is the use of nitrogen fertiliser? It's based currently on a finite resource, which is natural gas. BP tell us that globally, at the current rate of usage, global reserves are 51 years. and that usage is likely to go up even though we may well see more reserves discovered. So it's a very finite resource. You could produce nitrogen fertiliser from coal, but even coal reserves are only put at 118 years globally. So is there a long-term future for nitrogen fertiliser, or do we have to move towards using legumes? Now this is more controversial because I'm putting these on the table, we've got the scientists here, I'm happy to be shot down in flames. But Tom Misselbrook was looking, I think, at greenhouse gas emissions in the UK. Here I'm looking at greenhouse gas emissions globally, taken from a paper, and I've looked at a number of papers. Actually, this one is not out of balance with the others. Here we see that agriculture and land use change together account for about 22% of greenhouse gas emissions globally. However, the agriculture itself is only accounting for 7% of that. The biggest proportion comes from the land use change. And one of the things I wanted just to comment on is, and just to put this into perspective, is when people talk about livestock, inevitably they talk about the destruction of the rainforest or the destruction of the Cerrado in order to put cattle on there. But it's not really, that's a choice which someone is making, whether they put cattle or whether they grow crops. The real problem is coming in is because we've got a growing human population, and it's the conversion of virgin land to food production which is actually causing that change. It doesn't really matter whether or not it's arable or livestock. And finally, just looking at the methane sources, when you take methane from all sources, and this is globally, looking globally, we're looking here at ruminant enteric fermentation, is that saying 18%, something like that? And the manure mostly is related to intensive livestock production, including pigs. So it's not insignificant, I accept that. But if we're looking at 18% of enteric methane emissions, when actually agriculture is only responsible for 7%, in reality, the methane is only about 1.2% of total global warming. And I'd ask if we can actually produce food in a much more efficient way than that. Thank you.